Everybody, welcome back to another BrainMap seminar series. Today I have the great pleasure to introduce a very special person for me. He's not only an amazing scientist and a medical doctor, but he's a great collaborator, an all-time collaborator for me, and I need to say a great friend. And he's an MD. He got his MD at Columbia, if I am right, and he did his residency also at New York State. And came for his fellowship at the T-Ride, and then since he moved to the NGAs. He's both board in, let me get this right, he's certified for neurology and psychiatry, and he's got an appointment in both departments right now. His main interest is basically in using neuromodulation and neuroimaging to better characterize intrinsic corticolimbic networks and prefrontal cortex. So today he's going to talk about a super interesting project combining TMS, Transcranial magnetic stimulation, with fMRI, trying to find out what are the metabolic changes and the functional connectivity changes linked to TMS differences in both healthy volunteers and major depression. So I'm really looking forward to your talk. Thank you very much.
Thanks, David. Thank you very much. It's great to be here. So I'm going to talk. This is work I did actually as part of my K23 and finished it a couple of years ago. And so, it's advancing. Oh, slide's not advancing, David.
Yeah.
I just did that manually. So let me start by talking about what I call two different problems for cognitive and clinical neuroscience. The first problem is something that I personally think is a big issue, and that is that we have great people doing things at different spatial and temporal scales, but it's very hard to actually get coordination across those. So you see here in the little diagram here that it's basically depicting different temporal and spatial as well as topological and temporal scales. And it's really hard to have studies or investigations which actually link neural activity and processing and information processing across those. So I think that's a big issue. And it's particularly a big issue in association cortex. In terms of the connections in association cortex, it's a lot simpler in my mind in motor cortex because you have activity in motor cortex and then you have a readable output, right, in terms of movement of an arm or movement of a leg. But in association cortex, as can be seen here in this little diagram, has a great expansion evolutionarily compared to motor cortex, which means just intuitively that there's going to be a lot more connections, there's going to be much more of a sort of hub nature, there's going to be a lot of processing that goes through it. So this brings up, just as a very general problem, the dilemma between what happens locally in one brain region and how that impacts or how it's related in other regions. And getting back to the question of this processing across spatial scales, this is, I think, where we have to go and do a lot more work. And I'm not an electrophysiologist, not a basic researcher at all, but I think it's really cool to see studies that look at local field potentials or neuronal ensembles and relate that to characterizing brain networks. So the other specific problem is for those of us that do neurostimulation. And it's related to this, but our problem is that we do neurostimulation and we believe that we get some kind of effect as a result of the neurostimulation, whether that's an investigational effect or a clinical effect, but we don't really know how that is propagated. So we know that we are stimulating in a very focal area, let's say with TMS or with deep brain stimulation. So it could be that that's a non-invasive modality or an invasive modality. But again, we don't have a good sense of how that propagates. We don't have a good sense of the relationship between activity locally, where we're actually doing the stimulation, and that propagation. And that's really critical because we know, for example, those of us that work in the TMS clinic here, that TMS works for depression. There's a lot of studies that suggest that it does. And these are, these are a bunch of different FDA-approved devices as of this. I think the most recent one was 2018. So we know it works, but we don't know why. And anyone that tells you differently is probably fibbing a little bit. We think that it will modulate neural networks, specifically in cortical limbic and paralimbic areas, but we don't know the specifics of that. Okay. So one thing that we can do, and this is work by Thomas Paus, who coined this term perturb and measure. And the idea being that we could perturb a specific part of the brain and then measure what happens as a result of that. And so we did a study in 2011, seen here, where we stimulated the left inferior parietal lobule and were interested in changes in functional connectivity as a result of that stimulation. And we found that there were changes in the medial prefrontal cortex. The IPL is, of course, part of the default network. So we found changes within the network in the medial prefrontal cortex, as well as PCC, also in the default network. And then with a different form of stimulation, we found changes in the hippocampus. But again, the question is, what happens locally? What happens at the site of stimulation? So this brings my collaboration with David into focus, and this is a fused MR PET image, and this is work that I did downstairs in base 6, and this is just a picture of the MR PET scanner. Just want to quickly go over some advantages to combined MR PET scanning. So there's enhanced motion correction of PET images. There's better anatomical registration of PET images to anatomical structures. Did I do that? Sorry. There's lower radiation dose because there's no concomitant CT. There's attenuation correction is a big, big issue, but David has done a lot of pioneering work using pseudo CT, which can be created by the MRI to account for that, to ameliorate that. There are no physiological or state changes occurring between MRI and PET scanning. And one last thing that you can do, which I didn't do as part of the study, but you can do temporal modeling, and you can also look at the kinetics of receptor binding and link that to bold imaging, which I think is really exciting. So this was a study that was published about 5 years ago in the Journal of Neuroscience, and it was a combined MR PET study, and it involved just looking at having people do eyes closed and eyes open. And what you can see here, just very briefly, is there is this topographical overlap between the FDG, metabolic changes between closed and open, and the functional connectivity changes from primary visual cortex as well as within the salience network. So this is great. There's only been a smattering of studies that are like this, using MR PET and specifically looking at functional connectivity in terms of the MR portion of it. But again, it's still sort of a closed system. So everything is kind of happening spontaneously. And we thought that it would be really cool to do an external perturbation and do this, again, this kind of perturb and measure approach to see what happens. And again, like I said, this has the advantage for those of us who do TMS of finally kind of figuring out what is happening at the site of stimulation, which is a really, really open topic. I should also mention, by the way, it's such an open topic that for those of you who know stuff about TMS, you know that we basically will dose TMS based on a motor threshold. And we get that by doing single pulse TMS to motor cortex and seeing the least amount that will cause fingers to twitch on 50% of the trials. And it's been a controversy for years and years in the field of TMS about whether that's translatable to association cortices and to other parts of the brain. So we do a whole host of studies in DLPFC. In fact, arguably the majority of TMS studies as well as clinical applications are in DLPFC, and yet we continue to rely on motor cortex as a surrogate for cortical excitability there. Okay, so this is the overall general design of the study, and there are a lot of shortcomings, and I'll be the first to admit that, and I want to tell you about those and their limitations. But we start with a baseline MR PET scan. So people come in. We actually recruited about 20 healthy young people as part of the study. We also have a cohort of depressed people, which I'm not going to talk about at all today because I haven't gone into that data too much just yet. But we have them come in for a baseline scan, and then we have them return on a different day on 2 different days actually. The 2 days when they return are basically identical. The only difference is the site of the stimulation. So this is basically mimicking our design that we had back in 2011. But people get a regular fMRI scan with resting state functional connectivity, and then they get a fused MR PET scan after TMS, okay? So, and again, there's 2 different sessions, and the only thing that differs is the actual site of the stimulation. So TMS was delivered at 20 hertz. For those of you who are not as familiar with TMS, that is supposed to be excitatory. So again, getting back to motor cortex, we say it's excitatory because we do, there have been a lot of experiments in motor cortex where if you stimulate somebody with high frequency stimulation and then you do motor evoked potentials after that, you see that the motor evoked potentials become larger. So we glean from that and have for many years that high frequency stimulation is excitatory and low frequency stimulation, the opposite happens. So it's thought to be quote unquote inhibitory, okay? A big issue in this study that I wanted to bring up is the FDG injection. So that is the tracer that we're using as part of this study. It is F18, which means that it has a very long half-life, which means that we just started and have done a few subjects, I should say, with a C11 raclopride tracer with TMS. And that's a little bit of a nightmare because you have just a few minutes in order to do the TMS when it's actually active. The cyclotron sometimes doesn't actually create it, and sometimes it fails. But FDG is great. As you know, it's very stable, it's very reliable, it's transferred from an outside vendor, and you have a lot of time and leeway. But we did the injection, and this is a little bit arbitrary and up for debate, about 5 or 7 minutes before starting the TMS. So you can imagine someone arguing, well, you really should have done it while someone's getting the TMS. I should have had a slide on this, but I'm just going to draw it. The uptake, and David can correct me if I'm wrong, for FDG usually kind of follows this sort of asymptotic pattern. And so after about 40 or 60 minutes, that has flattened out and it's sequestered. So anything you do with TMS is not going to change the sequestration of FDG. So it's a really dumb idea to do it at that point. It's also not a great idea to do it way, way before the injection because there are debatable questions about whether the effects of TMS will just wear off by then. So this is, by the way, what's called an offline design, where you're actually getting your metric of interest without the TMS coil being on the head. So the duration of the effects of TMS is variable, and people quote about at least 20 or 30 minutes. Sometimes people will say it's half of the duration of the actual stimulation, but suffice it to say it's not permanent. So there is this kind of gray area about when exactly you should do TMS in TMS PET studies in terms of the uptake of the FDG. You really want to maximize the changes in FDG and attribute them to TMS. Okay, so as part of the analysis, the PET comparison that we made, and this is a little bit of a drawback also, is between the baseline and the post-TMS scan. And the reason that this is a limitation is that this could be, days in between this, in some cases even weeks, just because it's really hard to get, for those of you who scanned in base 6, it's really hard to get spots and, subject schedules and all that kind of stuff. So that is a limitation, but what you can't do is you can't do FDG and then TMS and then FDG, again, because of the half-life of FDG, because it's so long, so you just can't do before and after, unfortunately. So we kind of have to live with that. And then the fMRI comparison is sort of right before and right after, and that dovetails off of our previous studies where we just did pre and post. Okay, so where do we stimulate? So because we had patients in this study, it's, again, might seem a little bit arbitrary, but we were really interested in questions about depression, and Mike Fox, who many of you know, helped in the design of the study. And as some of you know, for people who are interested in TMS for depression, there's a lot of interest in what is the best site based on functional connectivity to do the stimulation. And Mike has really pioneered this idea that the negative correlations between the subgenual cingulate, which is, for those of you who work in depression, is thought to be this hub of networks in depression, that the maximal negative correlation with the subgenual cingulate is the best spot to go to. And we, myself and Tracy and Joanne, have adopted that in our clinic, where sometimes we will actually get a scan. So this is come into the clinic and actually try to hit the most negatively correlated spot. If anyone is interested, that's a little bit tricky because it always tends to be, the traditional spot, I should say, is this kind of 5 centimeter rule, which is 5 centimeters anterior to the motor strip. The negatively correlated sort of hotspot that people are leaning towards a little bit now tends to be a lot more lateral and a lot more anterior. And the problem with that is you have, in fact, we had somebody yesterday where the coil is kind of resting right on their eye, and that can be uncomfortable. There's a lot of the frontalis muscles there, temporalis muscle and everything. So you can get a lot of discomfort with that stimulation. But there is increasing work and increasing interest in targeting with TMS based on functional connectivity and specifically on anticorrelations. So we used an algorithm where we seeded the subgenual cortex. If Mike was here, he would say that you can't just do that because it's really hard to get, obviously, bolt signal out of that area, and there's a lot of variability. So we used an algorithm, which I'm not going to go into, in which we used cones that are distributed over the DLPFC, and we actually used a mask of the subgenual as a seed and got robust both negative and positive correlation. So those were our 2 targets. Again, on different days, somebody would come in, get scan, TMS scan, and only difference was the targets. And so I'm going to refer to this now and forever, even though it's a little bit weird, as negative and positive. Okay, so for the positive stimulation targets, which are here in black and are dorsomedial, if you prefer that term, and the negative, which are more dorsolateral. And as I said before, again, it tends to be this more anterior and lateral target. So these little circles, black and white circles, are just the different subjects, just representing where those targets are. So importantly, this is individualized, just as it is in the clinic. So we don't say, oh, well, I looked at the Yo parcellation and I looked at 1,000 subjects, and the maximal negative stimulation tends to be at these coordinates in left DLPFC. We're going to hit every person there. You could do that, and somebody could argue that that's actually better to do that, but we did it in this study and in the clinic on an individual basis. It's noisier, as those of you who do resting state functional connectivity know. You get much noisier maps. It's a little bit more difficult to interpret, but that's an open question for debate. Anyway, because I'm so interested in functional connectivity, and because I'm going to talk about changes in functional connectivity, it's important to sort of place this in the world of functional connectivity. And one of the parcellation that I used for this study is the Yo parcellation, which a lot of you are familiar with. I know there are many, many of them out there, and it's reasonable to say that I relied a little too much on the Yo parcellation, but this is the 7-network parcellation. So I just want to show you where these targets are situated in that, because it's very relevant. So on the 7-network parcellation, all of the sort of positive targets were clearly in default network. And all of the negative targets, a lot of them are in frontoparietal, which is this kind of yellow-orange, and then some of them are here in salience in the purple. But there's also the 17-network parcellation, which is a finer parcellation for those of you who know this paper. And interestingly, in this parcellation, almost all of our targets are in salience. And the reason that it's 17-network as opposed to 7 is that it subdivides many of the networks. So you have default A and B and salience A and B. And in this case, almost all of our targets are in salience B. Why do I mention that? Why am I harping on that? I'm going to give you a little bit of a take-home message of the study. It's all about default and salience. A little bit more so salience, but you're going to hear me mention those 2 networks in particular a lot. And I didn't necessarily go into the study expecting to find that, but that is something that we really saw. So again, just to hammer down this network topography issue, there are the different parcellations will differentially sort of divide up different regions, and I'm also going to be very interested in this region, which is dorsomedial prefrontal cortex, and I'll tell you why a little bit later. But in the 7-network parcellation, you see that the frontomedial, it's divided into this pure salience node and into this control MPFC node, whereas in the 17 network, it's more subdivided into the dorsal ACC of salience B, into frontomedial and salience A, and then there's this control, the small control sliver. Same thing with your frontal insular region, where there's kind of just salience and control, and then here there's A and B. To complicate matters further, and those of you, my friends from Harvard who have joined us know about this, this is work by Randy Buckner and Rodrigo Braga, where published in Neuron a couple of years ago, where it's not just that there is, they showed that it's not just that there is this fine-grained differentiation of cortical networks, but rather that every network has this parallel interdigitated representation in different parts of the brain. So if you heard Randy's talk a few weeks ago, I'm sure he talked about this, but it comes to the topic of a DNA and a DNB, where DNA and DNB are juxtaposed to each other. So again, it's not like DNA is in this part of the brain and DNB is in that part of the brain, but it's like AB, 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 AB. So it's really interesting, and it has some foundations in comparative anatomy and in track tracing, because, for example, network A is here in retrosplenial cortex, also in hippocampal formation, and in ventromedial prefrontal cortex. So I just show that just to discuss the complexity of targeting a network with TMS. And yeah, please, sorry.
Mark, I was wondering if you can go back one more slide.
Sure.
I'm still thinking about this. So you showed on the right that most of the little white circles fall in the little pink area, which is really quite a nice result. But I also noticed that it seems like most of the black circles follow the border and the boundary between those two, and that sounds like one heck of a coincidence. Is that coincidental, or does it actually seem to follow some sort of like a segregation boundary between the functional areas?
It's a great question. I think the best answer is I don't know. It might be that for, this is just based on the maximum. So we took, there's these different cones that Mike had, which represented all of DLPFC. I think there was 160 of them. And we just picked that one, I forget how big they were, I think 12 millimeters, the one that just had the biggest number for positive and negative. So it could be some, like you're saying, intersubject variability, where some people, their positive target is, I think what you're saying is just going to segregate. It's going to segregate into, the yellow here is default A, so it's just going to segregate into A or B, which is here. People, yeah, it's a great question. Okay, so some quality control metrics. Apo, what is in the back there, is working hard, and very grateful for this, to do TMS modeling post hoc, and that's that picture on the right. We did FC-FAST for this. I know for those of you who do functional activity, that's really old school. It's like ancient dinosaur type of stuff, sorry. But, ran it through an FC-QC pipeline. And then just to really push home the issue of subject motion, we took the best 2 out of 3 functional runs. There are 3 runs before and 3 runs afterwards, and heavily biased it based on slice-based SNR, as well as movements above 0.5 millimeters. We always took the first functional run after TMS, because I always wanted to capture that, because of course there's going to be some dissipation with time for TMS, so I always wanted to have that. In keeping with some of our other studies, the elapsed time between the end of TMS and the start of functional imaging is usually around, in our studies, about 5 or 6 minutes. So that means the coil comes off the head, and they're actually getting bold acquisition within 6 minutes. Again, this is stuff that Apo's doing, where there's some issues sometimes with NextStim, and what I want to do is just model the E field of all of the targets and just make sure I was hitting where I think I was hitting as best as we can post hoc. David, of course, doing the attenuation correction. All right, so the first thing to kind of go into that is very important is to just put a seed in the target for each person, and before you do any kind of TMS. And what's interesting, again, we're having this echoing of salience and default just so happens. So when we seed the positive target, we get a clear, the black outlines here are the default network, and the white outlines, this is the 7 network parcellation, by the way, are the salience network, and we just get this beautiful delineation of both the default network and, importantly, very importantly, of the anticorrelated network, so the network that is negatively correlated to the seed. It's not just any anticorrelations in the brain, right, which could be dorsal attention, maybe even visual, maybe frontal parietal, but it really seems to hug the contours of the salience network in particular. So that's an interesting thing that I don't know if people have looked at that much or if they've looked at enough, is just to think about, you know, we do all these parcellations of networks, but to think what are their sort of buddies, right? So for any given seed, we always talk about the positive functional connectivity, but what is the negative functional connectivity? There was a study by Udin and colleagues just showing that, you know, if you move a seed in PCC a little bit, you can get different negative correlations. So anyway, that's, I think, important. And then for the negative target, again, really getting salience, even though a lot of it in one of the things looked like frontal parietal control, but really getting salience and the negatively correlation, this is in the green-purple, is in default. And a little bit of interest here that the caudate kind of popped up in terms of the positive correlations for the positive target. Okay, so these are the PET results. And again, this is a lot of interest to those of us who do TMS. It's only 20 people, and actually it's a little bit less than that because we had to exclude a couple. But for the positive stimulation, even though this doesn't at all show up in this image, I'm sorry, but believe it or not, the metabolism actually went down. It was just barely significant. No peak correction, was literally like 0.09, 0.049, something like that. But what's weird is that it seemed to go down. And that, again, it's barely significant, but if you really believe that, if you think if we did more subjects and that bears out, that's super interesting to those of us who do TMS because, like I said, it's always, oh, high frequency increases cortical activity, increases metabolism, and then there's this suggestion that maybe that's not true. There's a lot of other parameters. It could be the duration of it. It could be the actual frequency. It could be that 5 hertz is different than 20 hertz, is different than 10 hertz, so on and so forth. Negative, and again, these comparisons are versus baseline. Negative versus baseline, boom, we see a nice, really nice increase in FTG metabolism at the actual target itself, and this is quite significant. Oddly enough, the positive targets seem to go down a little bit. And then the comparison of positive versus negative, it does seem clear that the negative target for negative stimulation was definitely more effective, definitely went up more than it did in positive stimulation. And there was this nice double dissociation there. This is just some covariance with the target. This is all FDG. So asking the question, are there certain networks that whose FTG is going to covary with FTG changes at the target? And it does seem as though with the positive stimulation, default B, using that network as a mask and looking at FTG in that network, did correlate with that, and with the negative target salience B did correlate with it. And this is just looking at different seeds of the different 7 networks and basically what's happening following positive, where we see an increase in salience. So I'm going to show you the actual pictures in a second, but it was not default, even though you're stimulating default. And then a huge increase in salience with the negative. And there is some here with control and the dorsal attention network. So kind of a big take home is that you stimulate DLPFC, left DLPFC, the salience network actually on the right, the contralateral side, kind of goes haywire. For those of you who are, if Marco was here, for those of you interested in pain, one of the things that kept me up at night for a while is, well, is this just a pain reaction, right? Because TMS can be uncomfortable, and particularly the negative stimulation. And I know this not just because I do TMS, but people said so. I didn't actually systematically acquire ratings of discomfort, but it definitely seemed as though the negative stimulation. So one concern is this just pain? Are you just getting increased metabolism in pain regions of the brain, many of which overlap with the salience network? But we didn't get it in posterior insula. We didn't get it in somatomotor cortex. So I don't think it's that, but anyway. This is just voxelized maps of the changes. So with positive stimulation, again, a lot of stuff is happening laterally here in this sort of perisylvian area. So default stimulation is causing a little bit of increase here at the sort of anterior tip of the frontoinsular cortex, and causing some in the posterior region of IPL. And then the negative stimulation is causing a huge increase at the posterior portions of the perisylvian cortex, in salience TPJ, as well as in the insula, and this is right hemisphere, but it did this is like right salience DLPFC, but trust me, it did it on the left. Well, I showed you. And then this is the difference between the two. This is a little bit weak, but still, I think of interest, and something we're pulling out a story from is that the metabolism changes definitely bigger with negative stimulation. With a positive, more dorsomedial stimulation, there was still nonetheless a little bit of changes here in this kind of dorsomedial prefrontal cortex. It wasn't specifically in default representations. It was a little bit in the salience B representation, as well as in the control PFC MP representation. But that's interesting, and if I have time, I'll try and go into why I find that interesting. So if frontal connectivity changes from the target, it's really important, right? It's where you stimulated. In the other study that I showed you that we did a few years ago, we saw big changes. Not so much here. You know, kind of wimpy. But what is interesting is that I think, and again, I promise you, if you liberalize this, it really does follow the networks that we stimulated. So in other words, and let me be more specific, for the negative stimulation, you actually see decreases with other elements of the salience network. For the positive stimulation, decreases with other elements of the default network. And so this gels with our prior study and gels with a couple of other studies, this idea that if you do high frequency stimulation, you decrease connectivity in the network that you're stimulating. Okay, some people have found increases, might intuitively make sense that you find increases. I can make up a good story why you see decreases. Specifically, you could very well just be introducing noise into the system and, you know, with the stimulation and not doing this coordinated increase across different regions. But this is, getting back to the beginning of the talk, this is one of the reasons why I think this is so important. Because you just have no idea what's happening on a network level and in a distributed manner. This is just showing different seeds that are not the target itself. But I divided this up just to show you something. So for the positive stimulation, and this is after we excluded a few people from motion. And before we did that, it was unbelievably dramatic. So the most dramatic thing that came out of this study was that for the positive stimulation, the anti-correlations, the negative correlations between the default network and the salience network tank. Okay, so if you just do, you know, and we did global mean regression for this, so that comes with all the caveats, but you just saw a drastic increase in networks that are usually negatively correlated. So for example, it could be that at baseline, the z-score is minus 0.6 and it goes to minus 0.1, or it even goes to 0, or even sometimes positive. And just this topographical pattern of drastic changes. You know, we've done a couple of TMS resting state studies, and usually you see, you know, a little bit of wisps here and there, but this is this really nice, take my word for it, topographical division based on the salience and default networks. And interestingly, again, getting back to my friend, the dorsomedial prefrontal / dorsal prefrontal, dorsal posterior prefrontal cortex, it seemed to be a little bit more, these decreases, these cross-network interactions were a little bit more relevant for this seed that was a little bit further back, as opposed to this one that's a little bit further forward, and that's divided in the 17 network parcellation. So bottom line, it's kind of, it changes in connectivity between the default and salience networks, and it's changes that are happening a little bit more posteriorly with the positive stimulation. So involving obviously PCC, it actually involved retrosplenial cortex a lot more than it did the rest of PCC, but also the perisylvian areas and big time this frontal medial. So contrast that with the negative stimulation and the functional connectivity changes that are resulting from that. So for the negative stimulation, again, remember we are stimulating, you know, right around here, let's say. But we're still seeing involvement of the prefrontal cortex. So in this case, we're seeding the actual subgenual cingulate, and we are seeing some increases in connectivity with sort of ventromedial prefrontal cortex, as well as with retrosplenial cortex. Here, seeding the orbitofrontal cortex, which is a big player in terms of the results for the negative stimulation. We're seeing these increases, again, with ventromedial prefrontal cortex. And then if we seed this area, which is the more anterior area here than here, in dorsal ACC, we're seeing these big changes with PCC and with orbitofrontal cortex. Okay, so just putting this into correlation matrices, the positive stimulation up here, so across salience and default, big changes. This could be driven by decreases within the salience network itself, because what you're seeing here, I should mention that blue, should have said this, blue are decreases in functional connectivity, so this is the change post minus pre. Red are increases in functional connectivity post minus pre. And this is just a legend showing that if it's bright yellow, then it's a p-value of that, and so on and so forth. So big cross-network changes between salience and default for the positive stimulation. Not so much across these limbic and perilimbic regions. And again, just to specify what those are, we're talking about ventromedial prefrontal cortex, dorsal medial prefrontal cortex. Got these seeds from the Drysdale paper that some of you may know, which is biotypes and resting network biotypes and depression, these specific seeds. This is the amygdala, the caudate, PCC, and OFC. Now you go to the negative stimulation, and you do still see a smattering of changes, cross-network changes. Interestingly, like I said before, they're most relevant with that dorsomedial prefrontal cortex. But you see kind of a lot of activity in terms of changes across these limbic and paralimbic regions. So and then this is just showing it across the entire brain, so the whole brain, and showing that, and with the 7-network parcellation, showing that we're really seeing the salience default action here, salience itself, and then here the limbic connectivity, and seeing that with the negative stimulation. So interestingly, this is these arrows represent the medial prefrontal cortex, ventromedial and dorsomedial prefrontal cortex. And the reason that I highlight them with these arrows is it kind of seems like they're doing something in both stimulation types. So just to show just topographically where these players are, I'm talking about like this frontomedial portion, dorsal ACC, dorsal medial prefrontal cortex, ventromedial, OFC, and subgenual. This is just a comparative anatomy representation of the different cellular architecture and between the macaque and the human. But then this is just a schematic of changes that occur, functional connectivity changes as a result of the positive stimulation and the negative stimulation. So for the positive stimulation, a lot of action here with PCC, which is in the default network, of course, as well as with retrosplenial cortex, and a lot of action with this frontomedial seed that's here. Whereas with the negative stimulation, still some action with PCC, some action with the dorsal ACC, which again is a little bit further anterior, and a lot of action in terms of OFC, orbitofrontal cortex correlations, ventromedial, prefrontal, and dorsomedial, including the caudate. So there was actually some significant changes between PCC and the caudate for the subcortical fans in the audience and the striatal fans in the audience, and BMPFC. Okay. Oh, and then one other big thing that we saw, because we have been kind of neglecting the subgenual, but one thing that we definitely saw with the subgenual was we saw increases with the left amygdala following negative stimulation. That kind of bore out across a lot of different analyses. So I started talking about the reason for doing these types of studies, and one of the things I was arguing was that it's really important to link different levels. And as I said, not an electrophysiologist, but what I think is so great about concurrent MR PET scanning is that you can get some idea of what is happening metabolically or at the receptor level, and then you can correlate that with the network level in terms of using resting state functional connectivity. So very exciting for me, but how did we, this is one of the most challenging analyses of the entire study. So how do we show that there is some link, first of all? And there's different relevance to the link. One is just this kind of general linking across levels. Another one could play into this whole idea of neurovascular coupling, just to show that, you know, metabolism does correlate with bold signal, that whole issue. But one way to do it is to just look at the topographic overlap. You're just kind of putting up two pictures and looking at if you're seeing the same patterns. You could actually overlap overlap the voxels themselves and check the correlation there. So this is for the negative stimulation, and it's not perfect by any means, but you are seeing a lot of, like I said, salience. In both cases, you're seeing FDG changes in the salience network, which is kind of the network stimulated, and you're seeing functional connectivity changes in the salience network. And then in this case, another thing you can do is say, how does FDG change as a function of the functional connectivity before TMS? So in this case, we're looking at the functional connectivity here to the default network before TMS, and then we're looking at the change in the SUV values, the mean normalized, whole brain normalized, I should say, PCC values, and we do see that as the functional connectivity goes down, we are seeing increases. So there's this inverse relationship. And the same thing, this wasn't true of the whole salience and default networks, but here is just looking at metabolism at TPJ as a function of its pre-TMS correlation with the network. And then the last thing, which I've spent months and months, and my kids say all you do is look at these stupid brains all day and get nothing done. And I'm just killing myself to try and pull this out. But one of the things we're trying to do is look at local activity, the FDG changes, and correlate that with the functional connectivity changes. And this really jumped out. This is for the positive stimulation. And when you look at FDG at the target itself, at the positive target, it correlates extremely nicely with the functional connectivity between this dorsal ACC region and the remainder of the default network. It seems kind of random, I know, but you know, we do see again this kind of default salience pattern, and we do see that we see this great correlation. So those are the different things that we have explored, that we're going to continue to explore to kind of link changes in FDG with distributed changes in functional connectivity. And again, this is important because if you're doing TMS or if you're doing DBS and you are delivering stimulation to a specific part of the brain, this is critical for where you actually want to go. It could very well be the case, and this does suggest this to some extent, that certain parts of the brain are much more sensitive, meaning that if you change their metabolism, you're really going to impact functional connectivity in the brain. Again, it could be a hub region, whereas if you stimulate other parts, it's not going to do too much. So you really want that kind of spatial specificity with it, and then you get into the whole issue of dosing, duration of stimulation, and the many, many other neuromodulation variables. Okay, so getting back to this, this is sort of, I'm just wrapping up here, but there's a lot of instances in my analysis where this part of the brain is popping up. And so just generally speaking, the medial prefrontal cortex, these are a lot of old studies, but is one of, if you believe in hubs and this graph network measures, a lot of these, by the way, there is some debate about the best way to characterize a cortical hub, but this is mostly based on your traditional metrics of degree and betweenness centrality. But a lot of these studies will actually cite the medial prefrontal cortex as a hub. PCC is a huge hub, but a lot of studies have cited the cortical midline in general as hubs of the brain. So again, it could be that there is something special about the dorsal medial prefrontal cortex when you do TMS to DLPFC in terms of its role in modulating functional connectivity. And lo and behold, this isn't a random study that I just pulled up to support my case. This is actually a pretty famous study. It's been cited a couple times, but Yvette Chalene came up with this idea of a dorsal nexus, and this was published in PNAS 7 years ago, 8 years ago, wherein there seemed to be a lot of functional connectivity converging on this specific region of the brain. So I kind of think of it as like a free agent, like it doesn't necessarily belong to a specific cortical network, but it has the ability and the responsibility to link itself to different cortical networks based on demand and specifically based on affective processing. So it's just interesting to think about this. And the only reason that I bring it up is just I kept seeing a lot of changes around this region. Okay, so just some conclusions, just wrapping up here, and then I'm happy to take any questions or any other discussion. So RTMS to the left DLPFC elicits metabolic changes and topographically really echoing the salience network. There's something very, very important about the salience network in terms of DLPFC simulation based on my results here. The functional connectivity changes induced by TMS to DLPFC do involve the stimulated network, but it's also, based on this data, it seems really important to take into account the negatively correlated system. Like I said, it's buddy. You can't just sort of look at the stimulated network in a vacuum because these effects are being propagated across its other network. The changes were characterized in terms of the positive stimulation, the more dorsomedial stimulation, with marked changes in default network salience connectivity, particularly laterally and posteriorly, whereas the negative stimulation target dorsolateral, which is what we do a lot for TMS in the clinic for depression, or more relegated to the rostral and ventral cortical midline structures, as well as subcortical, including the caudate and paralimbic structures. There are correlations that exist and that we're continuing to explore between metabolic and bold changes. So that could be explored as that Journal of Neuroscience study that I mentioned a while ago, just without any TMS at all with this data, but it can also be explored in an interesting way in terms of an external perturbation. So it's exciting to think about whether those would change, right? That if you do an external perturbation, it's not just the changes are exactly in the same direction, but maybe there is some divergence and some interesting topographical changes. And then finally, a better understanding of these correlations could improve circuit targeting for clinical applications as well as just understanding in general network dynamics and the interplay between cortical networks. So I wanted to acknowledge first and foremost Stephanie McMains, who's here in the audience, who has helped me tremendously with functional connectivity analysis, and I wouldn't be anywhere without her assistance, but also David for his amazing help with the PET, Oppo with the modeling. There is an amazing, if anyone hasn't scanned in basics, this is as good as it gets, these guys. They are just absolutely phenomenal and so helpful and professional and brilliant. And then I want to thank Mike Fox, Randy Buckner, Alvaro, and Ciprian for contributing to the design of the study. I'm happy to take any questions.